Yes, we did it. No. Okay.
Great.
Introduce to you.
Can I start, or there will be some introduction?
Just a moment, please. Our speaker is Agnieszka Majkowska. Pilip is from Institute of Nuclear Chemistry and Technology, Warsaw, Poland. She will present her presentation entitled "What is Radiopharmaceutical?" Yes, we are ready to listen you. Thank you.
Okay, thank you very much. Good morning, ladies and gentlemen. Good morning, everybody. First of all, I would like to sincerely thank the organizers, especially Zumrud, for inviting me to TÜBİTAK Summer School. It's really a great pleasure and honor to present my lecture today. As was mentioned, my name is Agnieszka Majkowska-Pilip and I'm professor at the Institute of Nuclear Chemistry and Technology in Warsaw, in Poland. In this talk, I will focus on radiopharmaceuticals, how they are designed, labeled, and used in nuclear medicine for both diagnosis and therapy. Oops. Okay. This slide introduces the concept of radiopharmacy. As you can see, radiopharmacy is a very complex field. It combines knowledge from multiple scientific disciplines like radiochemistry, coordination chemistry, organic chemistry, molecular biology, or radiobiology. Radiopharmaceuticals are medicines that contain a radioactive isotope, either as a simple radionuclide or as a molecule labeled with a radionuclide. They are especially designed for medical applications, mainly for the diagnosis of diseases and also for the treatment, especially in cancers. The preparation of a radiopharmaceutical usually involves 3 steps. First, we need to produce the radionuclide, for example, a nuclear reactor or cyclotron, or we can obtain it from the generator. Second, we synthesize the non-radioactive part of the compound, which can be a peptide, antibody, or small molecule. And finally, we perform the radiolabeling step, in which the radionuclide is attached to the molecule, creating the active drug that can be administered to the patient. Radiopharmaceuticals can be divided into non-targeted and targeted ones. Non-targeted radiopharmaceuticals rely on the natural distribution of the radionuclide in the body. Their uptake is determined by a physiological process or metabolic activity. And here, for example, iodine is widely used for thyroid diseases because the thyroid naturally accumulates iodine. Another example is radium-223, which localizes in bones and is therefore useful in the treatment of bone metastasis. Targeted radiopharmaceuticals are designed to selectively accumulate in diseased tissues. This is achieved by attaching the radionuclide to a targeting molecule, such as peptide, antibody, or small organic compound. These biomolecules bind to receptors or antigens that are overexpressed on cancer cells. For example, peptide receptor radionuclide therapy uses radiolabeled peptides to target tumors. Radiolabeled antibodies can be directed against cancer antigens such as CD20 in lymphoma, while small molecules can target specific transporters or receptors in tumor cells. Here you can see the general structure of a targeted radiopharmaceutical. And the radionuclide is bound to a chelator, which is connected to a linker and then to the biomolecule. The biomolecule, such as peptide or antibody, specifically binds to the receptor on the cancer cell. In this way, radiation is delivered directly to the diseased tissue, while exposure of healthy tissue is minimized. Now a little history. This slide shows a timeline of the discovery and development of radiopharmaceuticals. So it begins at the end of 19th century when Henry Becquerel discovered mysterious rays from uranium. And shortly after, Maria Curie-Skłodowska introduced the term radioactivity and defined new radioactive elements such as polonium and radium. In the 1930s and 1940s, iodine started to be used for diagnosis and treating thyroid diseases. Later in 1950, the first commercial radiopharmaceuticals became available. In the 1970s and 1980s, FDA approved new radiopharmaceutical technetium, indium, and thallium for myocardial imaging. From the 1990s, radiopharmaceuticals became more advanced with the introduction of FDG for cancer imaging and also radioimmunotherapy agents like Zevalin and Bexar. In recent years, a new area started with the approval of targeted radiopharmaceuticals such as Lutathera and Pluvicto, and also with alpha emitters like radium Xofigo. So this timeline shows how radiopharmaceuticals have evolved from the discovery of radioactivity to modern targeted cancer therapies. Here you can see a modified version of the periodic table, highlighting radionuclides that are used in nuclear medicine. Each color represents a different application. So blue indicates isotope mainly used for imaging with beta or gamma emissions. Green represent isotopes used for therapy, either with beta or alpha particles, and the orange color shows radionuclides that can be used in both imaging and therapy, often called theranostic isotopes. So here you can see a modified version of the periodic table highlighting radionuclides that are used in nuclear medicine. Each color represents a different application. Blue indicates isotopes used mainly for imaging with beta and gamma emitters. Green represent isotopes used for therapy, either with beta or alpha particles, and the orange shows radionuclides that can be used both in imaging and therapy, and they are called theranostic isotopes. For example, technetium-99, which you can see here, is the most commonly used radionuclide in SPECT imaging. Fluorine-18 is one of the most important radionuclide for PET imaging. Lutetium-177 and yttrium are widely used for targeted radionuclide therapy. And here actinium and radium are examples of alpha emitters that now are being investigated for cancer treatment. This slide summarizes the radionuclides that are used in nuclear medicine, divided into those applied for diagnosis and therapy. On the left side, you can see diagnostic radionuclide. For SPECT imaging, the most common isotope, as was mentioned before, is technetium-99, but there are also others like thallium-201, iodine, gallium, indium. For PET imaging, we use both organic radionuclides, such as carbon, nitrogen, FDG, fluorine with FDG, and also metallic radionuclides like copper, zirconium, yttrium, and the most popular in this group is gallium-68. On the right side, you can see radionuclides used for therapy. Beta emitters can be classified into hard and soft beta emitters. For example, strontium, yttrium, and rhenium are hard beta emitters. Softer beta emitters include such radionuclides like scandium, iodine, and here the most popular is lutetium-177. We also have Auger emitters, including radionuclides like indium, iodine, and lately the most popular terbium-161, which are very useful for delivering highly localized radiation at the cellular level. We have also alpha emitters like astatine, 211, bismuth, 212 and 213, and also actinium 225, which has become recently as one of the most promising isotope for targeted alpha therapy. The therapeutic effect depends on physical properties of the used radionuclides. So large solid tumors are treated with high energy beta emitters like yttrium. For small tumors, single cells, and tumor metastases, the best ones are alpha emitters and lately also Auger emitters. Beta emitters are characterized by the range in tissues up to 12 millimeters and linear energy transfer of 0.2 keV per micrometer. Alpha emitters, in comparison to beta, have a shorter range and high linear energy transfer, led to 100 keV per micrometer, resulting in high biological effect and also cytotoxicity. As we know, the biological effectiveness of alpha particles does not depend on hypoxia or also cell cycle. So in this way, alpha particles can make lethal double-strand breaks in DNA. The linear energy transfer of Auger electrons ranges from 1 to 23 keV per micrometer. Therefore, Auger electrons are similar to alpha particle and produce highly damaging effects in cells. Because the path length of Auger emitters is short compared to the size of a cell, they are minimally toxic to surrounding non-targeted cells, and it is possible to inject about 10-fold greater radioactivity of Auger emitters in comparison to beta particle emitters without toxic side effect. However, in the case of Auger emitters, to obtain a high therapeutic effect, it's necessary to deliver the Auger electron emitter to the cell nucleus in the vicinity of DNA strands. As was presented on lecture before me, the ionizing radiation may cause the formation of free radicals in the cell, which may damage the cell's DNA, or the damage also can be the result of direct impact, the hitting of an ion or electron on the DNA. This slide shows summary of FDA-approved radiopharmaceuticals, and according to the literature, current literature, 67 radiopharmaceuticals are currently approved, where 54 are used for disease diagnosis and 13 for therapy. Here, this graph A represents approval radiopharmaceuticals used in diagnosis and also in therapy of various diseases. And diagnostic agents are categorized here into 7 categories, and all radiopharmaceuticals are applied for, all therapeutic here, radiopharmaceuticals are applied for oncology. Graph B shows radionuclides used in PET, 37%, and in SPECT, 63%. We can, here in the PET, we can see fluorine, and mostly, and in SPECT, of course, technetium-90 now is mostly used. Graph C represents radionuclides used in cancer therapy, including iodine, yttrium, lutetium, and also others. If you look at the graph D here, showing numbers of approved diagnostic radiopharmaceuticals used in various diseases, cataloged by radionuclide, technetium here is the mostly used in clinical imaging for multiple diseases. Fluorine is used mainly in oncology and neurodegenerative diseases. And here the graph E represents targeting vectors for diagnosis and therapeutic radiopharmaceuticals. And here small molecules are used as the major vectors for radiopharmaceuticals discovery both in diagnosis and therapy. Currently, there are over 70 radiopharmaceuticals for therapy in clinical trials, and about 30% of them are based on what is important, are based on lutetium-177 alone. This table shows the beta and alpha emitting radiopharmaceuticals that have been approved by FDA for therapy. Examples include strontium and samarium for bone pain in metastatic disease, iodine for thyroid disorders, and yttrium-90 in forms such as zevalin or microspheres for liver cancer. More recently, we have seen the approval of lutetium-177 compounds such as Lutathera for neuroendocrine tumors and Pluvicto for prostate cancer. It's worth to mention that there is only one alpha radiopharmaceutical approved in 2013 by FDA. This is radium chloride, sold under the name of Xofigo. There are two main methods used for designing radiopharmaceuticals. The first one is direct labeling method, and in this case, the radionuclide becomes an integral part of the biologically active molecule and is directly incorporated into it. This approach is commonly used with non-metallic radionuclides such as fluorine in FDG or carbon-11 in choline. As is presented here, incorporation of a metallic radionuclide directly into a biologically active molecule may significantly affect in vivo behavior because of the integrated method is usually more complex and challenging when we are developing radiometal-based radiopharmaceuticals. So the second approach is the bifunctional chelator method or BFC method. Here the radionuclide is bound to a chelator which is then covalently attached to the targeting molecule through a linker. And this strategy minimizes the disruption of the biological activity of the targeting molecule and ensures that the radiopharmaceutical remains both stable and effective. Here you can see some of the most common chemical reactions used in bioconjugation. Bioconjugation is the process of linking a radionuclide through a chelator to a biomolecule. On the left side, you can see reactions between carboxylic acid or activated esters with amines, which form stable amide bonds. They are very frequently used because they are simple, these methods, and also robust. On the right side, we can see another important pairs, for example, isothiocyanates reacting with amines to form TURS, maleimides reacting with thiols to form thioethers, or alkenes reacting with azides in copper catalyzed reaction to give triazoles. This is so-called click chemistry, which is also very popular now. Another example is the reaction between trans-cyclooctane and tetrazine, which is very fast and efficient bioorthogonal reaction. And this reaction is often used in modern radiopharmaceutical development. There are four key components of bifunctional chelator-labeled radiopharmaceutical, which can influence the overall performance of the radiopharmaceutical. This is the radiometal, the chelator, the linker or conjugation method, and the targeting vector. Each of these components play an important role in ensuring that the radiopharmaceutical is stable, effective, and selective for the target tissue. To be clinically useful, a radiopharmaceutical must meet several requirements. It should have high radiochemical yield and form a very stable radiometal complex. It must target the diseased tissue effectively while clearing quickly from the non-target tissue and organs to reduce background radiation. Another very important point is the need to match the half-life of the radionuclide with the biological half-life of the targeting vector. For example, gallium-68 with a half-life of around 68 minutes is not suitable for antibodies, because they circulate in the body for several days. So this matching is crucial to optimize radiopharmaceutical for imaging and also for therapy. Many of radiometals used in radiopharmaceuticals have only one oxidation state available, like gallium, lutetium, in aqueous solution, and chelator selection for these radiometals is more straightforward. A few radionuclides like technetium, rhenium have multiple oxidation states, each with its own unique requirements for forming stable complexes. The lower oxidation states are softer Lewis acids, while the higher oxidation states are harder Lewis acids. Key factors that I considered when selecting a chelator for a particular radiometal include the complexation kinetics, thermodynamic stability, kinetic inertness, and in vivo stability of the radiometal chelator complex. This slide explains the factors that affect how quickly a radiometal chelator complex is formed. The most important parameters are the reaction time and the reaction temperature. I will speak about these parameters a little later. When working with radionuclides that have a short half-life, such as gallium, bismuth, long reaction times are a serious disadvantage because a significant part of the radioactivity can decay before the complex is ready. At the same time, high reaction temperatures can damage the biological activity of heat-sensitive molecules like, for example, antibodies. So we must be careful when choosing the reaction conditions. Ideally, we want the complex to form in less than 15 minutes and preferably at room temperature. Unfortunately, this is not always possible in practice. Finally, not only kinetic stability of the complex is important, but also the thermodynamic stability. The thermodynamic stability of metal complexes is usually determined by potentiometric titration and is reported as thermodynamic stability constant value, which is presented here, kml, where higher values than 18 indicate that this complex is more thermodynamically stable. The thermodynamic stability constants is also related to the forward and reverse rate constants, as indicated in the second equation. And the no carried added levels available, which we have it for most radiometals, nanomoles to picomoles range, kinetic stability here becomes more important than thermodynamic stability, because here the radiometal complexes are highly diluted in vivo. Kinetic stability means that the complex should not easily dissociate or exchange the metal with other ligands in the body. In this case, the dissociation rate constant K reverse is the key parameter. Finally, what really matters is the in vivo stability, so we know about it, and this is tested through biodistribution studies where we can see how the radiopharmaceutical behaves in a living organism. First First of all, complexes must be both chemically and biologically stable. It must also demonstrate biological efficacy and also show good biodistribution. So meaning high uptake in the target tissue and rapid clearance from non-target tissue. The chemical stability is dependent on the radiometal chelate, and thus the appropriate selection of chelator for radiometal is really critical. The radiometal chelate should have high thermodynamic stability and a very low dissociation rate, which means it should remain intact in the body. The choice of chelator is critical, as was mentioned, because even if we use the same radionuclide and the same peptide or antibody and changing only the chelator, it can significantly alter the biological distribution. It was confirmed by literature data. So this shows why careful selection of the chelator is essential for designing effective and safe radiopharmaceuticals. Radiometals available as the aqua ions are readily complex by polyaminopolycarboxylic acid based on chelators of various types and density. This ligand can be acyclic or microcyclic in nature and contain oxygen and nitrogen donor atoms. So firstly, I will focus on acyclic ligands. The TBAE is one of the earliest bifunctional chelators successfully used in the development of FDA-approved radiopharmaceuticals, including octreoscan, so indium DTPA octreotide used for SPECT imaging of somatostatin receptor, neuroendocrine tumors. DTPA rapidly form thermodynamically stable complexes with a variety of metal ions, as presented on the right side. However, most DTPA radiometal complexes, especially yttrium DTPA, are not in vivo stable due to radiometal dissociation and transhalation. Because of it, several DTPA analogs were also developed to improve in vivo stability. One such DTPA analog is 1B4M DTPA, where the incorporation of metal group improved the in vivo stability for indium and yttrium complexes if we compare to classical DTPA. This ligand, what is worth to mention, was also employed in the development of zevalin, which is a radiopharmaceutical used for radioimmunotherapy of non-Hodgkin's lymphoma. CHXA DTPA is currently considered the most suitable DTPA analog for complexation, yttrium, lutetium, bismuth. And at the bottom there is DFO ligand. This is a standard chelator for zirconium and forms strong complexes with this PET radionuclide. The main feature of acyclic ligands is that they form complexes very quickly, which is an advantage when working with short-lived radionuclides. However, they are often less stable in biological fluids, which can lead to release of the radionuclide in vivo. This is why acyclic ligands are useful in some applications, but for many radiopharmaceutical, more stable are macrocyclic ligands. So one of the most popular and widely used chelator in radiopharmaceutical chemistry research is DOTA. Generally, DOTA is a suitable chelator for many radiometals like indium, yttrium, scandium, holmium, terbium, actinium, and also others. This ligand is typically conjugated to biomolecules for one of its carboxylic arms. DOTAGA as DOTA derivative contains one decarboxylic acid pendant arm, which can be conjugated to the biomolecule. And generally, the complexes with DOTAGA are more stable. Conjugation to a biomolecule can be also accomplished through p-isothiocylates the cyanobenzyl group incorporated into DOTA chelator, and this is PSCNBN DOTA bifunctional ligand, which you can see here. TCMC is a DOTA analog designed mostly for complexation of lead isotopes and contains 4 amide pendant arms. Compared to lead DOTA complexes, lead TCMC complexes were shown to be more resistant to dissociation at lower pH, less than 3.5. Here this table shows the stability constants of various metals with chelators. As you can see, the stability constants for DOTA chelators are a little higher than a little higher in comparison to DTPA acyclic ligand. This slide presents examples of other macrocyclic ligands, which are very also important chelators in radiopharmaceutical chemistry. The first ligand, NOTA, has the smallest binding cavity in microcyclic polyaminocarboxylic acid chelators, which make it ideal for smaller radiometal ions, especially gallium. As present here, the stability constants for gallium is 31, whereas for copper is 21.6. NOTA generally is currently the gold standard for gallium with fast kinetic complexation at ambient temperature, what is important, and also excellent in vivo stability. Other ligands like NODASA, NODAGA, or NETA contain 1 decarboxylic acid pendant arm. Theta is a microcyclic chelator that has been primarily investigated for copper. Similarly to DOTA, theta has an octadentate coordination, but with a larger ring size. And copper theta complexes were not really stable in vivo due to transhalation of copper. Therefore, new ligand CBTE2A was synthesized to improve the kinetic inertness and in vivo stability. However, in the case of this ligand, one main disadvantage is that it requires really harsh radiolabeling conditions, 1 hour at 95 degrees, which is not ideal for antibodies. Because direct attachment of the complex to biomolecule can result in a reduction or loss of its biological activity, we can use linkers to distance the complex from the biomolecule. Linkers are also used to modify the properties of the radiopharmaceutical, for example, its lipophilicity. And finally, they can also modulate the pharmacokinetic properties of the overall compound. A very important requirement is that linkers must remain stable under physiological conditions. In some special cases, cleavable linkers can be used, but in general, they should not interfere with the specificity or binding affinity of the targeting vector. As presented here, a linker can be a simple hydrocarbon chain increasing lipophilicity, or a polypeptide chain like polyglycine, polyserine, increasing the hydrophilicity of the radiopharmaceutical, of course, leading to fast excretion through the renal urinary system. And many studies show that certain groups of linkers have an influence on the biodistribution of monoclonal antibodies labeled with indium and technetium. The radiopharmaceutical in the structure also contains a biologically active molecule, which can be peptide, monoclonal antibody, or nanobody affibody. Peptides are relatively small molecules with a molecular weight around 1.4 kilodaltons, and they allow for fast clearance from the body and good receptor-specific targeting. Monoclonal antibodies such as trastuzumab, which is presented here, but also other monoclonal antibodies like pertuzumab, panitumumab, are very large molecules, around 150 kilodaltons, which bind very specifically to tumor-associated antigens. Because of their size, they have slow pharmacokinetics, which makes them suitable for use with long-lived radionuclides, like, for example, zirconium-89 for imaging. Nanobodies are much smaller, about 12-15 kilodaltons, and they represent only the smallest functional part of the antibody. And thanks to their size, nanobodies combine high specificity with, of course, faster blood clearance compared to full antibodies. So in summary, depending on the application, we can choose between small peptides, large antibodies, or intermediate nanobodies, affibodies as our targeting vector. Here I present different classes of peptide use as targeting vectors in radiopharmaceuticals. So the first and most widely used are somatostatin analogs such as dotatate or dotatalk, which target somatostatin receptor 2. When labeled with gallium, lutetium, actinium, they are applied in imaging and therapy of neuroendocrine tumors and also for therapy of astrocytomas. Another important group are bone-basing analogs, which target GRPR receptors. These applications, they have applications in prostate, breast, colon, and lung cancer. LGD peptides target integrins, exendin 4 binds to the GLP-1, neurotensin analogs target NTSR1, and substance P analogs bind to NK1 receptors, and they are investigated for glioblastoma therapy. Finally, PSMA targeting peptides are highly specific for prostate specific membrane antigen, and they are labeled with gallium for diagnosis, lutetium or actinium, and applied in prostate diagnosis or therapy. Here I present examples of monoclonal antibodies that are used also as targeting vectors in radiopharmaceuticals. The first two, ibritumomab, duoxetan, and tusitumomab, which target the CD20 antigen, they have been applied in the treatment of non-Hodgkin's lymphoma. Trastuzumab, pertuzumab target HER2 receptor, which is overexpressed in breast and gastric cancers. These antibodies can be labeled with radionuclides such as zirconium, copper, indium, lutetium, also actinium. Cetuximab targets EGFR and is used for colorectal as well as head and neck cancers. Bevacizumab targets VEGFR receptor. And finally, also J591 is a monoclonal antibody directed against PSMA for imaging and therapy of prostate cancer. Also, in preparation of radiopharmaceuticals, stabilizers play a very important role. Ascorbic acid is one of the most common stabilizer. It works as a radical scavenger or antioxidant, meaning that it reacts with radiolysis products before they can diminish the radiolabeled compound. In this way, it helps to maintain the radiochemical purity and stability of final product. Usually, stabilizers are added at low concentrations because too much ascorbic acid, for example, could interfere with labeling chemistry by binding to metal ions. And also from my experience, also ascorbic acid can interfere in some biological experiments like MTS assay. It can interfere with this MTS compound. Depending on the type of radiopharmaceutical, different stabilizers are used, what are presented on this slide. Now I would like to shortly focus on radiolabeling with technetium, which is the most widely used radionuclide in diagnostic nuclear medicine. And in fact, about 80% of all routine diagnostic applications rely on technetium. As all of you know, it's produced from molybdenum generators and has very favorable properties. It's almost a pure gamma emitter, making it ideal for SPECT. Because technetium has multiple oxidation states, it offers great flexibility for labeling a wide range of molecules, including small compound peptides and proteins. Radiolabeling of peptides and proteins with technetium can be performed in different ways. One approach is direct synthesis, for example, by adding hexahistidine tag at N-terminus, which binds technetium for a two-step reaction. And another widely used approach is conjugation with a bifunctional chelator, like in example Heineck. However, because Heineck alone cannot fully saturate the coordination site sphere, additional coligands are added like EDDA, trypsin, to stabilize the final compounds. Here you can see the structure of Heineck TOC, which after labeling is used for diagnosis of neuroendocrine tumors. The second important radionuclide used for PET imaging is gallium-68, who is produced from a germanium gallium generator. It decays mainly by positron emission with high energy, and it has a short half-life of about 68 minutes. For labeling, the most common chelators are DOTA and NOTA. DOTA has a larger cavity, so it requires higher temperatures, usually around 90 to 100 degrees Celsius for complexation. In contrast, NOTA can form stable complexes even at room temperature. When evaluating gallium-68 from the generator, this gallium is obtained in hydrochloric acid, and the solution is then buffered, typically with HEPAS, phosphate, or ammonium acetate. A critical factor here is keeping the pH below 4, otherwise gallium hydroxide will precipitate, which reduces the radiochemical yield. Some of the most important traces include gallium DOTA for neuroendocrine tumors, gallium PSMA 11 for prostate cancer imaging. Now I would like to focus on crucial parameters influencing radiolabeling efficiency, especially pH and buffer system. Different buffers are used for labeling depending on the radionuclide and of course biomolecule. For example, acetate buffer is the most common for radiometals like lutetium and yttrium. HEPAS buffer is widely used for gallium chemistry, while phosphate buffer is often applied for proteins and antibodies. Carbonate buffer can be used in radioiodination. Citrate helps stabilize technetium complexes, and tris buffer is most suitable for actinium-225. So the choice of buffer is critical. It must be free of competing metal ions such as iron or zinc, which would interfere with labeling because the stability constants iron DOTA is really very high. It's higher than with yttrium or lutetium. It also has to be biocompatible, especially if the final product is injected into patients. And also buffer concentration must be carefully controlled, since too much buffer can also compete with the chelator. In addition, also some buffers like HEPAS are not approved for human use unless they are completely removed, so quality control here is really essential. Also pH itself plays a crucial role. It controls metal ion speciation and chelator binding efficiency. It ensures biomolecule stability and prevents unwanted site reactions like hydrolysis or colloid formation. Another key parameters that strongly influence radiolabeling are temperature and reaction time. Temperature accelerates the complexation between radionuclide and chelator. For example, DOTA requires relatively high temperature, often around 90 to 100 Celsius, to achieve efficient labeling. However, such high temperatures can also damage sensitive biomolecules, especially proteins and antibodies, which require much softer conditions. Reaction time is also critical. Ideally, it should be, as I mentioned before, it should be kept short between 5 and 30 minutes. And this minimizes radionuclide decay losses, which is particularly important for short-lived radionuclides like gallium or bismuth. Shorter reaction times also make the clinical workflow more efficient, while overly long reactions can increase the risk of radiolysis and product degradation. In practice, small molecules and peptides can tolerate higher temperatures, while proteins and antibodies need lower temperature and longer incubation times. To address these, more reactive chelators such as NOTA or HBED can be used, which allow labeling at room temperature. And another approach is pre-labeling strategies, so where the chelator is labeled first and then conjugated to the biomolecule. The final part of my speech will be focused on nanotechnology and research performed in my institute. As you know, unfortunately, some radionuclides that are very attractive from the medical point of view, as presented here, like alpha emitters, actinium, bismuth, astatine, lead, radium. Some, like radium and astatine, do not form stable chelate bonds. Therefore, it's difficult to bind them with a biomolecule. Moreover, some of them not are very stable in vivo. But these difficulties are met by nanotechnology, which allows for the preparation of, for example, radioactive gold nanoparticles, adsorption of astatine on the surface of gold nanoparticles, or immobilization of radium emitter into the structure of the nanozeolite. These issues are dealt with by the team at the Institute of Nuclear Chemistry and Technology in which I work. Due to the time, I will present only research about the use of radium-223 in nanotechnology. So in recent years, radioactive nanoparticles have been increasingly used in both diagnostic and medical therapy. Their main advantage is that each nanoparticle contains at least hundreds of radionuclide atoms, but in the case of conventional radiopharmaceutical, the targeting biomolecule is labeled only with one or few radioisotope atoms. This is very important, and this allows for a much higher activity in radiotherapy and is particularly important in Auger electron therapy. Nanoparticles are characterized by high surface area to volume ratio, efficient radionuclide loading and labeling, also straightforward easy synthesis, and enabling the production of constructs with diverse physicochemical characteristics like shape and sizes. In nanomedicine, there are a lot of nanostructures like liposome, quantum dots, GOT nanoparticles, magnetic nanoparticles, micelles, carbon nanotubes, dendrimers, and others. The future of nanoparticles in medicine is also related to their multifunctionality, consisting in combining diagnostic and therapy in one radiopharmaceutical. And moreover, nanomedicine also allows for the simultaneous use of a number of therapeutic methods, for example, chemo and radiotherapy in one nanoparticle, which is extremely important due to the drug resistance of cancer cells. It's also worth notice that nanoparticles themselves have the ability to accumulate within a cancer tumors through the so-called passive transport, defined as increased permission and retention. And this is related to the leakage of the tumor blood vessels into which nanoparticle easily penetrate from the circulating system. There are also nanoparticles with the attached vector, which bind to the receptor or enter the interior of cancer cells by endocytosis. And this is active transport, allowing increase the effectiveness of the therapy and reducing the cytotoxicity to healthy cells. And one of the very promising alpha emitter which can be used in targeted alpha therapy is radium-223. Why this radionuclides? So firstly, radium-223 emits 4 alpha particles along its decay chain with a total energy of 28 MeV. A short range of this radionuclide could make this emitter very useful in the case of critically located tumors like glioblastoma. Another important aspect of radium is its half-life of 11.4 days. And this longer half-life in comparison to, in example, short-lived radionuclides would make radium more efficacious in the case of larger glioma tumors, where sufficient intratumoral distribution could be achieved, leading to effective tumor cell kill. Radium could be also used for distance metastasis. Moreover, as I mentioned before, radium esophago is already used in patients suffering from prostate cancer that has spread to bone, and the medical results are very good. But unfortunately, radium, like calcium, is a member of the second group periodic table and form very weak complexes with the ligand. So in this case, the labeling of biomolecule by classical way is impossible. On the other hand, some zeolites like zeolite A exhibit high affinity for heavy cations of the first and second group like cesium, francium, radium. Zeolites are aluminosilicates where negative charged zeolite skeletal is neutralized by cations, mostly sodium. So it's possible to easily synthesize nanoparticles of zeolite with diameter from 7 to 50 nanometers. So in our institute, nanozeolites were synthesized by hydrothermal method. The synthesis was prepared in the following molar ratio reagents. The temperature was 100 degrees and the reaction time was 96 hours. Size distribution and shape of nanozeolite particles were investigated by DLS, STEM, and SEM analysis. It can be seen from the SEM and TEM images that the nanoparticles are in size between 50 and 170 nanometers. However, the DLS analysis shows that the suspended in water nanoparticles are of the size between 120 and 160 nanometers, but this is, of course, hydrodynamic diameter. And in the next step of this studies, the PEC linker, 2000 kilo Daltons comprising the silan group at one end and also enhanced ester at the second. And the vector, here we use substance P511, were used for the synthesis of silan of this bioconjugate. The synthesis as presented was performed in DMF and for 48 hours, and the final product was purified by HPLC method and lipophilized. To synthesize radiopharmaceutical exhibiting affinity to NK1 receptors on the glioma cells, the active fragment of substance P from 5 to 11 amino acids was conjugated to the nanozeolite. And this modification was performed in 96% of ethanol. Sealant PEG agent was attached to the surface of nanoparticles by siloxane bonds formation. High concentration of hydroxyl groups on nanozeolite surface allow attachment of significant amount of this bioconjugate. The obtained product was characterized by DLS and thermogravimetric method as presented on this slide. In the next step, our bioconjugate was labeled with radium by exchange of sodium cations, which are present in the channels, and interstices of nanoparticles. So, and with current labeling conditions, radiobioconjugate was obtained in high radiochemical purity, more than 99%. The stability studies of these bioconjugates were carried out in saline solution, PBS, and human serum at different time points, and they were measured by radiometric analysis. And here, after 19 days of incubation in 37 degrees, around 95% of complex remained in the solution of saline and PBS. Radiobioconjugate was also stable in human serum. Human glioblastoma tumors exhibit NK1 receptors on the surface of the cells. So the binding affinity was determined by replacement of radioiodinated substance P by our nanozeolite. Here we calculated the IC50 value, which was 14.53 micrograms per ml, and this value was similar to the literature data. Also, we performed cytotoxicity studies, MTS assay, and here, due to different amounts of radioactivity added, a different percentage of viable cells was present. Cell viability also decreased with increasing incubation times. When the cells were treated with almost tenfold higher doses, only 30% of them were observed as metabolic active. The same experiment also was performed with radioresistant glioblastoma stem cells. And after 4 days of incubation with 10 kilobecquerels of radiolabeled nanozeolite, around 54% of cells were detected as viable, so almost 2 times more than in the case of normal glioblastoma cells, suggesting, of course, the resistance of glioblastoma stem cells in comparison to normal glioblastoma cells. As you can see, hot control with the use of radium did not show any cytotoxicity. So to summarize, the nanozeolite was successfully prepared by hydrothermal synthesis. The modification of the surface makes these nanoparticles stable and dispersed in water, which was confirmed by zeta potential measurement. The radiolabeling purity was over 99%. This radiobioconjugate retains good affinity, the NK1 receptors. Although this radiobioconjugate was found to be highly cytotoxic, not only towards the glioblastoma cell lines, but also to radioresistant glioblastoma stem cells. And targeted alpha therapy with this radiobioconjugate is promising approach for treatment of glioblastoma multiforme and also warrant further investigation in vivo. We are approaching to the end of this lecture, so I would like to shortly summarize the radiolabeling procedures. So no single chelator will be ideal for all metals or all bioconjugate. This is not always direct correlation between thermodynamic stability and in vivo stability at the radiotracer level. Acyclic ligands, due to their rapid formation kinetics with metal, can be used in combination with monoclonal antibodies or their fragments, where elevated temperatures cannot be used in labeling process. For other biomolecules where elevated temperature can be used for labeling, it's better to use microcyclic ligands that exhibit inert bonds with metal cations. Important to match the appropriate chelator to both the radiometal and also the biological target. Regarding to the nanotechnology, which I mentioned, there are also some limitations with radiopharmaceuticals. As you know, radiolabeled nanoparticles tend to accumulate in the liver and spleen, limiting their effectiveness for tumor targeting. Therefore, they can be only administered by intratumoral injection or into the cavity after surgical resection. resection. So thank you for your attention, and if there are any questions, I'll be happy to answer them.
Fidan nasıl yapalım? Gel. Hı? Gel.
Thank you for the talk. I have one question.
Yeah, please.
I wonder what is the relationship of the size of the molecule when you prepare the radiopharmaceuticals? I mean, at one side, clinician wants to use a radionuclide because of the decay properties of that probably, and on the chemistry side, trying to prepare pharmaceuticals. So what is the relation? I mean, you try to get a stable radiopharmaceuticals.
Yeah.
So you try to make an efficient chelate or the peptide, but it would be very big, right? Or I mean, in on paper, it's kind of a long molecule structure, let's say. So what is the dependence on this? The efficiency of this molecule in when one patient take into or the clinical side. I mean,
right?
So, in clinical, mostly there I use peptides, yes, the small biomolecule because of the pharmacokinetic, yeah. So if we design radiopharmaceutical, as I presented, so the chelate is very important, yes, because sometimes there can be the dissociation in the body, yes, and also it's very difficult sometimes to compare in vitro results, stability studies with in vivo. So I think that generally the short peptides are much better than monoclonal antibodies at this moment. And the stability is really very important. But I don't know if I catch your question, because could you, did I answer or?
Explain it maybe in a right way.
Okay.
So I think it's okay. So I got my
It's okay
answer.
for you because
Yeah. Okay, thank you.
you're welcome.
Agna, thank you very much.
Thank you.
Everything is okay.
Yeah.
It's pleasure to join us today. See you soon.
See you soon. Thank you very much, and have
You're
a nice
welcome.
summer school. And
Okay, okay. Thank you.
thank you.
Bye
Bye bye.
bye. Bye bye.